review. Okay, so just one quick thing. I have test graded, and so at the end of class, I will hand those back. I'll stop about 10 minutes early, and I'll pass those back. I will post a key up to the test on D2L, so you can take a look through there. One thing I would suggest you double check is that if you do not mark darkly on the Scantron, sometimes the Scantron only picks up a few of your answers. It will not pick up all of them. So I would encourage you to go back and take a look at the key and look at your Scantron, or not your Scantron, your test, just to see if you think there are some that may have been missed. And if you think so, let me know, and I will go double check your Scantron just to make sure that the machine did not miss any, or if it did, give you those points back. So that's just something to keep an eye out for. Let me get these back and I'll try to post up the thing. Okay. But with that, we are going to launch into talking about these specific G protein second messenger systems that I kind of ran up to the beginning of at the end of the hour on Friday, but didn't really get into. So the first one we're going to talk about is this one up the top, the Adenylyl cyclase cyclic AMP. And so before I get really deep into that, let's talk about a few abbreviations that we're going to be tossing out. First off, I'm going to refer to Adenylyl cyclase as AC. Just by the name, does anyone know what kind of molecule that probably is? A what? An enzyme, yeah. It's an ACE at the end, so it's an enzyme. And then CAMP, or cyclic AMP, is just cyclic adenosine monophosphate. So it's like ATP, but we take the two P's off. Sounded weird how I actually, after it came out of my mouth. And then we cyclize it. I'll show you a figure of what that looks like in just a moment. But cyclic adenosine monophosphate. And then pKa, what I'm talking about here is protein kinase A. Again, an enzyme. A kinase specifically is an enzyme that is going to phosphorylate another enzyme. So we're going to stick a phosphate on another protein. I'm going to talk about, just kind of like an aside, I'm going to talk about protein kinase A as if it is one individual enzyme, but really protein kinase A is a family of enzymes. So just kind of keep that in mind. Okay, so cyclic AMP, this is the structure of cyclic AMP, just kind of harkening back to what we had seen before. Again, you have your nucleotide here, or your adenosine right here. It's hooked onto a ribose. And then you have your 1-phosphate, but unlike the like a normal AMP, where it's just hooked up to this or bound to this oxygen right here, it's also bound to an oxygen down here, and so this makes it cyclic. Okay, so let's talk about this pathway very specifically, the cyclic AMP adenylyl cyclase. So just recalling, actually, before I go back, let me just really fast recount the basic G protein system. So let's just come back really quick before I do that, and let's just recount the basic G protein system. And that we're getting a message in. We have some sort of signal molecule that's going to bind to a G protein coupled receptor, a G protein GPRC, G protein coupled GPCR. There we go. Coupled receptor. Yeah, a G protein coupled receptor. Me and like. I can't even think of the word. Not analogies. They are. What's that? Abbreviations. That's still not the word. Those are good. What's that? Initialisms. Initialisms? No, that's not the word I'm looking for.
Not acronyms.
What's that?
Not acronyms.
That's not what I'm looking for either. Or acronyms. No. I don't know. Never mind. Okay, so but that is going to then, when the ligand binds to the G protein coupled receptor, it's going to bind to a G protein. Specifically, the G protein, in kind of like its state before it binds to there, has a GDP bound to the alpha subunit, and then that alpha subunit has a high affinity site for G protein coupled receptor. It's going to bind to that. In the course of binding to that, that is going to cause the GDP to pop off and a GTP to bind, and then also the alpha subunit to come away from the rest of the G protein. That alpha subunit is then going to go on, bind to some effector. It could activate it, it could deactivate it, but then it's this effector that then creates the signal on the inside of the cell. So this is how we get something changing on the inside of the cell without our signal molecule ever actually entering the cell. It's actually kind of relaying a message onto something, the effector, that is then going to carry out making that message on the inside of the cell. Okay, so that's the basic bare bones of how that works. And then essentially what this ATP, or excuse me, this alpha subunit, this will generally hydrolyze back to a GTP or GDP, go back to bind with the beta gamma subunit, and then come back to the beginning, and it can go all over again. Okay, so let's go back specifically to the adenylyl cyclic AMP system. We have the typical thing here where we have some ligand bind to a G protein coupled receptor, which is going to bind to a G protein. The alpha subunit is going to pop off. In this case, the effector that is going to bind to and in turn activate is the enzyme, the membrane-bound enzyme adenylyl cyclase. Adenylyl cyclase, what it does is it takes ATP and converts it to a cyclic AMP. So it's just going to sit there and continue to do that, just making cyclic AMP from ATP. The cyclic AMP then, in turn, can go and bind to a protein kinase A and activate the protein kinase A. The protein kinase A, in turn, being a kinase that phosphorylates something, generally a protein, in this case an enzyme, is going to phosphorylate an enzyme. So here we have some enzyme. I'm not going to name what that enzyme is at the moment. But that enzyme becomes phosphorylated, and generally, again, this is not exclusive, but generally that'll become activated, and that will turn some substrate into a product, as enzymes tend to do. Okay. So, just to recap that whole thing, because it's a lot of steps. It's kind of crazy. Like, why in the world have all these steps? It's like a Rube Goldberg machine. You are setting up all these steps to do something simple, like, I don't know, when your alarm goes off, it like goes down and makes eggs for you. Okay, maybe not exactly that. Although the product could be scrambled eggs. I don't know. So the G protein binds to G protein coupled receptor when there's a ligand bound. Alpha subunit activates adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic, ATP to cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A will phosphorylate enzymes, and then a substrate then is turned into a product by that enzyme, and then this product then is the thing that we want. Can anyone think of any good reason why you would do it in this complicated way? Why not just have adenylyl cyclase make the product? Why do it this way? Yes, that's one good reason. It's easier to regulate. Why would this be easier to regulate?
So many steps, you can stop it anywhere.
Yeah, there's a lot of steps. You could jigger with this at a lot of different places. That is the scientific term for regulation, is jiggering. Not really. I'm joking there. Any other good reasons why this could be important? To do it this way instead of just having adenylyl cyclase making the product. Location. Okay, so that could be important because you want this not to necessarily be right here, right? Is that kind of what you're thinking? Yeah. In some cases, that is an instant. That is a instance. That is a thing that happens. I'll just say it that way. But that's not the one I'm thinking of. But a good thought. Anything else anyone's thinking of? What's that? No.
Why
Yeah, why not? Why have all these steps? Why not just make the AC or whatever effector this is here? Wouldn't be adenylyl cyclase in that point. But why not just have the effector just make the product that you want?
What if you had CAMP somewhere else in the cell and you make it so you wouldn't have to have it start from there? Like you could start from a different point.
You could start from a different point, yeah. That's a good point. Yeah.
I feel like if you have a really big structure that does like something that's super important right away,
Yeah.
that structure somehow gets inhibited like unnaturally, then that's like a whole process that doesn't get done. But if
Okay.
you have a bunch of little steps, it's easy to regulate. Yeah, you can inhibit in any of the steps, but it doesn't like rely entirely on just one structure.
You can argue the other way that it makes it more liable to break because if you break any of the parts, then the whole thing breaks. You could go that way, kill it.
It's like more universal. So like you like in one cell you have the same like first like 3 steps in making a single candy, but then depending on what protein kinase you have in each cell it like makes a different product.
Yes, that's a really good. Okay, so kind of I'll call that customization. But another thing I'm going to call that is divergence of signal. Okay. Some other, yes.
Would it be like a small amount of ligand is needed for a thing? You only need a small amount to have a huge reaction.
Yes. Amplification. Let me talk about how this works, because that's a very good insight. But might not be readily apparent how in the world you get signal amplification from this. Here's how that happens. So the first thing that we have is that we have a ligand binding to the G protein coupled receptor, right? That G protein coupled receptor doesn't necessarily just activate one single G protein. It can activate several G proteins. Those G proteins then activate some adenylyl cyclase, and then the adenylyl cyclase makes lots of cyclic AMP. So for every first messenger that you have in this system, you get about 1,000 cyclic AMP protein kinase A couplings happening. So that's just for one ligand that you have bind. Then for every one of these, essentially for every activated pKa that's been activated with the cyclic AMP, you're going to get 100 of whatever that enzyme is downstream of that phosphorylase. So now we have 100 of those enzymes activated. And then enzymes work really great because you can just make, depending on what the enzyme is, though, you can get about 10,000 product molecules for every enzyme that you activate. And these are rough because, again, we're talking about kind of a generalized system right now. And so these are kind of roundabout numbers that are close to the average for each of these steps. What this means, if you multiply this out, if we have 1,000 pKas, which are each going to make about 100 enzymes, which are each going to make about 10,000 product molecules, this means that you have like a billion-fold amplification. Wait, did I do that right? Yeah. That's a lot. So for one ligand that you have bind to the exterior of the cell, you can get somewhere on the order of 1 billion of the second messenger or the final product that you want on the inside of the cell. So you have this incredible ability for signal amplification. Let's talk about something else, though, too. I like the other one here Kayla talked about. You can have a lot of different things that are then activated by protein kinase A. So, in different cells, you have different things that can end up happening. And actually, let me go to this slide first. and then come back to that site. So you actually have a bunch of different things that pKa can phosphorylate. You don't necessarily need to know this list. The point is that this is a small subset of the things that pKa can phosphorylate. Also, glycogen synthase, phosphorylase kinase A, protein phosphatase 1, pyruvate kinase, liver tyrosine hydroxylase, acetylcholine receptor delta, protein phosphatase inhibitor 1, hormone sensitive lipase, phosphofructokinase, just a ton of things all over the place. Can be activated or potentially deactivated by protein kinase A. And not all of these are in the same cells, but some of them are. So one of the things that you can have happen is just because you activate protein kinase A, it can activate several different enzymes. And so actually you have the signal not just going, I showed it here. Come back here. I showed it back here as just a linear sort of, we got protein kinase A and we phosphorylate enzyme and we get some sort of product. But in reality, what's happening is protein kinase A will do this enzyme and phosphorylate another enzyme and another enzyme and another enzyme. And so you actually get several different kinds of products being made. It's not just one kind of thing. And that's what I mean by signal divergence, is that from one ligand, you can get several different actions. That signal can diverge into several different products. But it also allows some sort of customization per cell, depending on what kind of enzymes you have present that are being expressed in that cell. You can get different kinds of products made. And so, depending on what cell you have and what kind of enzymes you have present, what different kinds of pKa substrates you have, a different ligand here can get you very different products made in that cell, having therefore very, very different effects. And so if we go back and we look at the different effects of different cells, like, and in adipocytes, you can get enhanced lipolysis. In skeletal muscle, increasing, and again, you don't need to know this exhaustive list. This is an example. But the effect of this in skeletal muscle can be increasing glycogenolysis, decreasing glycogenesis, and increasing glycolysis. Here's a funny thing, though, is look at this. Increasing glycolysis in skeletal muscle, but in hepatocytes, in liver cells, it's actually decreasing glycolysis, doing two completely opposite things in different cells. And it depends on the needs of those specific cells in that specific tissue. And generally these tend to be, well, actually let's just read down these. So in adipocytes, you're increasing lipolysis with breakdown fats. In skeletal muscle, you're increasing the production, glycogenolysis, or the breakdown glycogen. You're decreasing making glycogen, and you're increasing glycolysis, the ability to make energy. In cardiac muscle cell, you're sequestering calcium in your sarcoplasmic reticulum. So you're actually taking calcium out of the sarcolemma there, or the interior of the cell. In hepatocytes, we're also increasing the breakdown of glycogen and decreasing the production of glycogen, but now we're turning down glycolysis, and we're increasing gluconeogenesis, which is the making of new glucose from non-glucose molecules. In principal cells, this is actually in your kidneys. We have aquaporin 2 expression, and then smooth muscle, we actually have relaxation happening. So, can you think of what all these random things are doing? Like if you look at all these together, what kind of a big effect could this have on the whole organism? Okay, sympathetic nervous system, but what does that do? Well, what is the global response that has? Yeah, fight or flight. Essentially, all of this together is getting ready to mobilize energy. Hence, we are breaking down glycogen, we're breaking down lipids, and we're making new glucose molecules. We are getting energy ready to get used or created in skeletal muscles. And other places that don't need to work hard, like your liver, if you're running away from a bear, you don't need your liver working hard. That's like that's not a big thing that you need. Well, at least not making a lot of energy, so we're actually going to turn down glycolysis in your liver because that's not a big priority at that moment. The interesting thing here, and actually what this does, is this aquaporin 2 expression in the principal cells, we're in the kidneys now, we're actually increasing the reuptake of water. So we're actually not making as much pee at the moment. And smooth cells, like smooth muscle cells here, we get relaxation. Kind of what's going on here is smooth muscle cells, especially in your gut, are trying to move food through your gut, and they get really active, like when you're resting and relaxing, and your guts start like really moving. But if you're worried about running away from a bear, for instance, you don't really need your guts moving. Like, let's just pause on the whole digestion thing for a little while and get away from the bear first. And so we have relaxation on smooth muscle. It's kind of interesting because we have this one pathway that has very different effects in different places of the body, but altogether they have the effect of coordinating one complete response. And this is all made possible because, again, back here, we can have divergence of signal here, pKa activating different things here, and depending on what these enzymes are in a particular cell, it can elicit different responses. If we did not have a signal cascade, if we just had adenylyl cyclase making some product, we couldn't do that. That wouldn't be possible. So we don't have that ability to customize these different responses based on that if we don't have this signal cascade. So just recapping there. Signal cascades allow for several things. Number one, you can have lots of different control coming down here. So control. I'm going to add that up here to the... But also you can get signal amplification. One ligand now can make a ton of products. We can increase the number from the ligands to the products like 1 billion-fold in that neighborhood. Now also we can customize what the responses are depending on what cell type we have. Or we can actually have divergence of signal where we have multiple things happen. One signal, but multiple different things being made. Okay. Another thing that's interesting here is also involved with this is a protein called AKAP, which is a kinase anchoring protein. And it's interesting. What it does is it holds pKa in a specific location in the cell. So we can hold on to pKa and keep it anchored somewhere. And this is good because then we can also hold other enzymes in close relationship to the pKa. For instance, maybe whatever enzymes is going to phosphorylate. Another really interesting one that often gets held by AKAPs are phosphodiesterase. A phosphodiesterase is a type of enzyme that will break down cyclic AMP. Why would you want to have an enzyme that breaks down cyclic AMP really close to your pKa? In what way? Say that again one more time. Yeah, you want to turn it off eventually. Not only do you want to turn it off eventually, you want to be able to turn it off quick if you want to. This is actually something that comes up a lot in physiology, cellular physiology, but then also whole organism physiology, is you have a lot of things to break down signal molecules in the same places that you are producing those signal molecules. This happens all the time. In your synapses, in your neurons, or between your neurons, you have a ton of enzymes to break down neurotransmitters. Like, why would you have them in synapses? Because you want to be able to turn off. You want, as soon as you take your foot off the gas, essentially, you want that signal to turn off right away. You don't want lingering on for a while. So phosphodiesterases in this case, as soon as we stop making cyclic AMP, we want to turn off the pKa's. We don't want them to just keep going after we stop making the cyclic AMP. So we have phosphodiesterases that chew up the cyclic AMP, break it down, and then that signal can stop really quickly. And there's about 50 different known AKAPs, or more than 50 different AKAPs. So there's a lot of different specific AKAPs that will hold both protein kinase A, but then also other enzymes close to it, other proteins close to it, that allows it to be in close proximity to the enzymes that it works with, essentially, and so we can make that more efficient. This is part of the regulation that we were talking about earlier, for enzymes is by physically holding them in a location close to other enzymes, we can control what happens to the products and what happens to the substrates really nicely. Okay. Are there any questions on the protein kinase A cyclic AMP adenylyl cyclase pathway? Okay. Let's talk about the next one, calmodulin, which is a protein that just takes its name for calcium modulated protein, calmodulin. And this is just an enzyme that is going to bind for calcium ions and then it becomes active. Like, you give it calcium and it's happy and it can go, it's like it's coffee. And it's actually not all that big. It's only somewhere in the neighborhood of about 148 residues, which isn't that great. That's not that large. It's kind of small. This is an image of calmodulin. I always like putting it up there because it looks like a happy enzyme. Like, it looks like it has its two eyes, and it's like, yay. Hang on, so that's calmodulin. These black balls are the calcium ions. So let's look at how calmodulin can work its way into a G protein second messenger system. Here again, we are beginning with like the exact same beginning. G protein coupled receptor binds to a ligand, activates G protein, alpha subunit goes off, and in this case, the effector that's going to affect is actually a calcium ion channel that is going to open up, and then we have calcium flooding into the cell. In this case, we actually have the calcium bind to calmodulin. This can do a couple different things. One of the things is that calmodulin oftentimes activates phosphodiesterase to break down cyclic AMP. And so this can be used to shut off other systems. So if we break down cyclic AMP, one of the things that will happen is you can shut off your protein kinase. So this can have an inhibitory effect. In addition, calmodulin can directly activate protein kinase Bs, some protein kinase Bs, or some protein kinase Cs, which then go on to do a very similar thing to what we saw protein kinase A do. Phosphorylate an enzyme, generally activate it, though sometimes deactivating it, and then making some product out of the substrate. It's fairly similar to the adenylyl cyclase cyclic AMP, but we're just doing this slightly differently. And it gives you an idea here that this doesn't necessarily need to make things. The effector could be letting things in. The next G protein second messenger system is a phospholipase 3, IP3, and DAG system. Phospholipase here, just PLC, will break down PEP2, which is phosphatidylinositol 4,5-bisphosphate, or just PEP2, into IP3, which is inositol 1,4,5-trisphosphate, and a diacylglycerol, 1,2-diacylglycerol. Okay, so there's a lot of really big words. Phosphatidylinositol. What do you think that is, based on the name? Phosphatidyl something or other. Any ideas at all? Yeah, Hector? Well, phospholipase C in this case is the effector. The phosphatidylinositol battle. What's that? I heard you say something? Yeah, phosphoglyceride. So this is just a phosphoglyceride. And we happen to have a couple phosphates stuck onto it, more than you would normally expect there. So remember what we have in a phosphoglyceride. You have your glycerol. You have a couple fatty acid tails. bound to a phosphate to your polar head group. In this case, what do you think the polar head group is? Inositol. So we have inositol here. We have inositol. But this is 4,5 bisphosphate. So actually on this inositol, we have a couple phosphates on there. A couple more phosphates. That's really all we have. And what we're going to do is make inositol 1,4,5-trisphosphate and 1,2-diacylglycerol. So can anyone like imagine what would we do to this to get those two kinds of molecules, you think? What is it?
Just separate the 1,3 phosphates and the
Like this?
Just bring it there, right?
Yeah, just bring it there. So now we have inositol trisphosphate, and we have a 1,2-diacyl 2 hydrocarbons glycerol. That's really all we're doing. So just to show it again, here's our 2 fatty acid chains. Here's our residue of glycerol to the fatty acid to the inositol, our 2 phosphates on there. Phospholipase C is just going to break this right here. So then we have a 1,2-diacylglycerol and this, which is the 1,4,5 inositol trisphosphate. Okay, so just knowing what this is and knowing where the rest of this is occurring, where do you think this is before we catalyze it? Yeah, it's in the membrane. It's just sitting in the membrane like a normal, hunky-dory, happy phospholipid until phospholipase C comes along and chops its head off. So sad. So, again, here we have a G protein coupled receptor, G protein again, activates the G protein, alpha comes up, and it then affects phospholipase C. In phospholipase C, we have our PIP2 here, just as one of the normal membrane one of the membrane phospholipids, just doing its thing. Phospholipase C comes here, or phospholipase C breaks off the inositol polar head, or the inositol bisphosphate polar head. It's taking the phosphate from the phosphoglyceride with it, and now you get IP3 with the inositol 1,4,5 trisphosphate. And the diacylglycerol just hangs out there it doesn't leave the membrane. We just chopped this polar head off, but the diacylglycerol still is perfectly happy staying in the membrane. So it just does that, stays staying in the membrane. But the DAG, the diacylglycerol, then can activate a protein kinase C. However, the protein kinase C needs one other thing to fully become active. It needs calcium. And so the IP3 actually goes to the endoplasmic reticulum, and the IP3 will bind to a calcium channel. That calcium, or the calcium channel in the endoplasmic reticulum, opens up and releases calcium. Actually, turns out the smooth endoplasmic reticula have a lot of calcium in them. And it opens that up, releases the calcium. That calcium can then go to a protein kinase C, which I'm just pointing it down here because it's easy to like get the arrow over there, but again, we're up here in the membrane. And then the protein kinase C is fully active with your calcium plus your diacylglycerol, and it can now do its thing. I didn't put it up here, but what do you think protein kinase C is going to do? Yeah, phosphorylate enzyme. Just going to do its normal thing. It's just going to go phosphorylate enzymes, like a self-respecting protein kinase alphabet soup, and then that enzyme can then make things. Or perhaps in that case, could deactivate and it stops making things. Okay, so we've gone through this and we've talked a lot about what alpha is doing. You might think, well, beta gamma is just like the sad sack second and third wheel. They just happen to be there whenever alpha wants to come around and be around, but then like alpha's always leaving and doing stuff with effectors. But it turns out that the beta gamma units aren't as quite satisfactory just sitting around the house as you might think they are, waiting for alpha to come back around, pay attention to them again. In this case, actually, beta gamma units can have their own effects. And they can activate or inhibit all sorts of other intracellular effectors themselves. It turns out that we weren't first looking at the beta gamma subunits. The alpha was easier to study, so that's the one. That is the subunit that has ended up being the one that we more focus on. But beta gamma units can also have their own actions on effectors. It's not only the alpha subunits. So some of the things they can do, they can activate potassium channels in the cell membrane, and open that up, and potassium will flush. Do you think that go into or out of the cell? Out of the cell, yeah. Generally out of the cell. We can inhibit calcium channels. We can affect adenylyl cyclase with beta gamma units in some ways. Sometimes they can also affect phospholipase C in some ways. I say affect because it depends on the cell if they're going to activate or inhibit. So I just say affect. So beta gamma units, I haven't talked about them much, but no, they got a life too. Okay. Hey. Okay, it's my wife and kids. I had to. I want to say hi. Anyhow, okay, so let's move on. So that was our G protein and second messenger systems. However, that's not the only type of second messenger systems that we have. We actually have a plethora of others. G proteins are the most well known, but we do have some others, like the protein receptor kinases, which operate a little bit differently. We do not have a G protein for this particular second messenger system. It is just different. We have a protein receptor kinase. And these can be involved in a whole bunch of things, but they tend to be involved in long-lasting effects. Long asking something? I don't know. So things like cell growth, development, differentiation. Usually we're working through gene regulation, but not always. In fact, some of the canonical protein receptor kinases are not working through gene regulation, but as we've studied more of them, we found many of them actually do. There's two different protein receptor kinases that we're going to be talking about. The first of them is the receptor tyrosine kinases. There's a lot of these. We have over 50 different known receptor tyrosine kinases in humans. And you probably guess what they do. They're a receptor, and they're a kinase, which means they're going to phosphorylate a thing. And what do you think they phosphorylate? Like, where on the protein do you think they phosphorylate a thing? On the tyrosine. They're going to phosphorylate a tyrosine. See, it's simple. I've heard astrophysicists say that. Like, all of our naming schemes in biology are really complicated and hard. Well, they're all very simple. Like, it's a red dwarf star. No, that's not a thing. Never mind. A brown dwarf star. This is simple. This is, well, kind of simple. It's not quite like red giant or brown dwarf simple, but it's simple-ish. And so what they're going to do is they're going to phosphorylate a tyrosine residue on a receptor tyrosine kinase. They phosphorylate each other. Isn't that nice? And once that happens, once a receptor tyrosine kinase phosphorylates another receptor tyrosine kinase, a protein with a specific kind of domain called an SH2 domain, it can dock with the receptor tyrosine kinase once it's been phosphorylated, and then it's either going to become active or inactive, depending on what it is. One of the most common of these is, not the most common, the most well understood. One of the very first ones we began studying is the insulin receptor. Okay. So we're about 10 minutes out. So I do have this really awesome graph here of insulin receptor, but we'll save that for Wednesday. I'll hand back tests now. Oh. And once you do your test, feel free to do whatever.